Okay, so thank you to the organisers for the invitation to talk today, and especially in a session about immunotherapy, and I don't work on immunotherapy at all, but rather what our work focuses on in the Botel lab is genomics, and now we're starting to look at how genomic changes in tumours perhaps interrelate with the immune response. Okay, so I don't need to tell this audience that patients with high-grade serous ovarian cancer have variable responses to primary treatment. So right through from primary refractory, where patients' CA125 serum level doesn't return to normal, to primary resistant, where patients relapse within 6 months after finishing their primary treatment. And you've heard Liz talk about acquired resistance patients, so we've got a large interest in that in the lab. And my work is really focused on not just primary sensitive patients, but those primary sensitive patients that do really well. So this work looking at exceptional responders really started quite a number of years ago when David Botel, Anna DeFazio, Paul Harnett and Kath Alsop noticed that there are these patients that have exceptionally durable responses to primary treatment. And they fall into these two groups. So long progression-free survivors, where you have an extended period of progression-free survival, and then these patients that continue to respond to platinum-based chemotherapy over several cycles. Over several lines of treatment. And some of these patients also become long-term survivors. And we see this additional pattern of patients that, despite having residual disease, they have this extraordinary response to chemotherapy and, in fact, never relapse. So this led us to the question that in these good responders, there's still these differences in the clinical histories of these patients and how they respond to treatment. So this suggests fundamental differences in tumour biology. So we first set about defining exceptional responders. So we looked at all of the epithelial ovarian cancer patients in AOCS and the Westmead Gynae Biobank, and decided that we would do a survival outlier analysis, but restricted to the 780 or so patients that were initially recruited to AOCS consecutively over the first 5 years of the study, and only include those patients with high-grade disease, advanced stage, and that were all treated with primary platinum. And to cut that long story short, we came up with a definition of survival greater than 10 years for long-term survivors, which fits with what most people say in the literature. And long progression-free survival was greater than 3 years. And these patients, we also stipulated that they had to have residual disease so that we could ensure they weren't cured surgically. And the multiple responders had 3 or more complete responses to platinum treatment. So when we applied those strict criteria, we identified 96 exceptional responders. And you can see only a subset of these are multiple responders, so these patients are rare, and not many of them go on to become long-term survivors, unfortunately. But the long progression-free survival patients, the majority of those do go on to become long-term survivors. So we wanted to ask 2 main questions of this unique set of patients. So what are the molecular features associated with long-term survival and exceptional response to chemotherapy? And do these patients have a more favourable immunological response? And today I'm hopefully going to hint at what we're looking at now is this interplay between the genomic changes and the immune response that we're seeing. So the obvious place to start was germline mutations in these patients. Do BRCA1 and BRCA2 germline mutation carriers account for most of these patients? And they really don't. So compared to an unselected cohort of high-grade serous patients, which have a rate of about 17% of germline mutations, we see that there is an enrichment in the 3 exceptional responder groups, but sort of underwhelming and not statistically significant. So we extended this analysis to tumour DNA to look at somatic changes in the extended homologous recombination DNA repair pathway. So looking at a panel of over 30 genes associated with HR DNA repair pathway or genes of interest in ovarian cancer. Plus we also looked at BRCA1 and RAD51C promoter methylation. And we found this extraordinary enrichment for pathogenic mutations or alterations in exceptional responders. So 73% of these patients, when we grouped them together, had an alteration in the HR repair pathway. And this compared to 41% in the unselected TCGA cohort when we considered just pathogenic mutations in the same panel of genes. And when we break the exceptional responders down into their subgroups and the long progression-free survivors even further into less than 1 centimetre residual disease on the left and greater than 1 centimetre residual disease on the right, we see a further enrichment for alterations in these genes. And this suggests perhaps this is related to chemosensitivity, and especially when you look at the multiple responders who have the largest frequency of mutations in the pathway, but the numbers, of course, in that group are small. But I guess the other observation here is that there's still a lot of long-term survivors that are wild type. So what else might be going on? So an obvious question, and from everything we've heard today, we know that CD8 positive lymphocytes are important in survival. So we looked at immune cell infiltration in these patient subgroups. So perhaps unsurprisingly, we found using immunohistochemistry across tissue microarrays compared to the unselected high-grade serous patients, those that had long-term survival or long progression-free survival had a large enrichment for CD8 positive lymphocytes in the tumour epithelium as well as in the stroma on the right. And I'll come back to the immune system in a minute. So one of our other questions was, aside from the HR repair pathway and known genomic defects, what novel genomic changes are associated with these patients? And so Liz described earlier our work on ICGC, and one of the sort of incredible findings from this study, because we're using whole genome sequencing, we're able to identify structural variants. And so the 80 patients with the green bar there, that's all primary tumours. The range of structural variants, this is total number of structural variants per tumour, ranges from about 50 right up to 700 in some tumours. And when we map these back to the genome, we see that down the bottom here, these red bars are where structural variants are mapped to known tumour suppressors. So we found for the first time that aside from point mutations, tumour suppressors are also inactivated by gene breakage. And within this cohort, we actually had 19 cases that were also classified as exceptional responders. So a group of long progression-free survivors in purple and then multiple responders in pink. And we saw this incredible enrichment for inactivation of RB1, mostly by gene breakage, but also by homozygous deletion, which had been described previously. But we'd never before seen such an enrichment for mutations in RB1. And we went to see if this manifested at the protein level, and using immunohistochemistry, we can see an example on the left of a tumour with RB1 wild type, so no change, and you retain RB1 expression beautifully. And this is an example of a tumour with a structural variant, so you can see complete loss of the protein from the tumour cells. And this holds up across the rest of the cohort, for which we had genetic information matched to the protein. So when we looked at the exceptional responders compared to our control group of unselected high grade, we see in the blue bars there an enrichment in long-term survivors and long progression-free survivors for RB loss, and further, when we break down into residual disease status for the long progression-free survivors, we see an enrichment in those with greater residual disease. So we did wonder whether this, again, is a biomarker for chemosensitivity. So what are the mechanisms underlying enhanced survival in patients with RB1 deficient tumours? So looking at the literature, we know that RB1 has a well-established role as a tumour suppressor, but so far not a lot of work has been done in high-grade serous cancer. We know that RB1 is a negative regulator of the cell cycle, so in normal cells, RB1 binds to E2F, which is a transcription factor, preventing it from activating its downstream targets. And E2F's downstream targets, or the best characterised, are those genes required to progress through cell cycle. So in tumours at least, loss of RB1 is thought to confer a proliferative advantage. So in other cancers, this has been linked to chemosensitivity. So potentially losing RB1, you get upregulation of proliferation, and perhaps that is affecting your sensitivity to cytotoxic chemotherapy. So is this what we see in high-grade serous cancer? So this work is still ongoing, but early indications are that perhaps this isn't the case. So this is work being carried out by Florina and Jess in the lab, and Florina has generated high-grade serous cell line models where she can induce knockout of RB1, and this is using a hairpin strategy. And in this wild-type cell line, there's no difference in sensitivity to cisplatin or paclitaxel, and she also found the same in this BRCA1 mutant line. So the data we have to date suggests that perhaps RB1 knockdown doesn't directly affect chemotherapy sensitivity, but we are carrying out this work in additional cell lines and also using CRISPR knockout, which we think might reflect what we see in the patients more closely. And going back to the tumour data that we have, I'm not sure if this is projected very well, but on the left side, this is comparing, so in the blue, RB1 lost tumours to those with RB1 retained, and quantifying Ki67, which is a proliferative index. And there is sort of a higher median Ki67 score in the RB loss tumours, but sort of an underwhelming difference, and there's certainly no statistical significance between these two groups. And if you, on the right here, divide tumours by low Ki67 and high, there's no difference at all between RB1 loss and RB1 retained. So at least with the data that we have to date, it looks like enhanced survival of patients with RB1 loss can't be contributed to a hyperproliferative phenotype. So what else is going on? This brought us to sort of look back at the immune system. And again, this work is ongoing, but we, to date, haven't been able to show an incredible enrichment for CD8 positive lymphocytes in RB1 loss tumours, but there is a higher level. But I will also say that we're going forward and looking at a bunch of other tumour markers, immune markers and immune cells across our exceptional responders cohort. But I will highlight some work we've just started to do, which is to compile our existing whole genome data with our RNA-seq data from ICGC and look at the difference in gene expression between tumours with RB1 loss and tumours with RB1 retained, and see if we can see any patterns in the genes that are differentially expressed. So I identified those that were more abundant, or more or downregulated in the RB1 loss tumours, and performed a gene set enrichment analysis to look at overrepresented pathways. And what I found, at least enriched in the RB1 loss tumours, is a few different statistically significant pathways. So the top the top one being E2F targets. So that being sort of reassuring that we're seeing that, because RB1 loss, as I mentioned earlier, you expect E2F to then be able to upregulate transcription of its targets. But the second and third pathways are interferon alpha response and interferon gamma response, so potentially indicating that in RB1 deficient tumours we see upregulation of immune markers. And in these pathways we do see genes associated with antigen presentation. So this is something we're exploring. So in summary, we've shown that exceptional responders are associated with germline and somatic mutations in the HR pathway. We've also shown for the first time that RB1 loss is associated with exceptional responders, and also, as probably expected, we see an enrichment for CD8 positive lymphocytes. And what I haven't actually shown is that in our long-term survivors, every single long-term survivor has at least one of these features. Usually there's an overlap, but every single long-term survivor has at least either high CD8, or HR repair pathway deficiency, or RB1 loss, or a combination. And obviously this is something we're looking into further. But just to say that in terms of RB1 loss and what might be causing a better response in these patients, we certainly haven't been able to show that it's due to a hyperproliferative phenotype, but perhaps there is an enhanced immune response in these patients. And so what's next? We're obviously performing more comprehensive immune profiling. We know from other work that especially B cells and plasma cells are associated with superior outcomes. We're performing more functional validation. So as I mentioned, looking at RB1 knockout as opposed to knockdown, and whether or not this is associated with upregulation of different or downregulation of different immune markers. And also would be comparing RNA-seq data in cell lines to what we've seen in our human tissues. And also just to finish by mentioning that we're currently involved in MOCOG, which is a large international study looking at not just the immune system and genomics across long-term survivors, but also integrating epidemiology, which I think will, all three aspects are going to be very important to long-term survival. And so in that study, we're looking at up to 1,800 patients, so 600 long-term survivors compared to short-term survivors and medium-term survivors, and looking at all of these three aspects. So I'll finish there and just acknowledge everyone that's contributed to this study, and thank you for your attention.